Hello guys, you're welcome to Alicia's Made Up. We'll be discussing sex-linked or X-linked disorders. So X-linked disorders or sex-linked disorders are X-linked disorders, and almost all are recessive. Several genes are located in the male-specific region of Y chromosomes. All of these are related to spermatogenesis. Males with mutation affecting the Y-linked genes are usually infertile. Males with mutation affecting the Y-linked genes are usually infertile, and hence there is no Y-linked inheritance. This accounts for a small number of well-defined clinical conditions. The Y chromosomes, for the most part, is not homologous to the X, and so mutant genes on the X do not have corresponding alleles on the Y. Therefore, the male is said to be hemizygous for X-linked mutant genes. So these disorders are expressed in the males. So generally, what this thing is saying that X-linked disorders are usually expressed in the males. They are expressed in the males. Females usually carriers, but males they express this disease, express this condition. So other features that characterize the X-linked disorder: an affected male does not transmit the disorder to his son, but all daughters are carriers, like I said. So males express the disease, while females are carriers. Sons of heterozygous women have, of course, one chance in two of receiving mutant genes. The heterozygous female usually does not express the full phenotypic changes because of the paired normal allele. Because of the random inactivation of one of the X chromosomes in females, however, females have a variable proportion of cells in which the mutant X chromosome is active. Therefore, it is remotely possible for normal allele to be inactivated in most cells, permitting full expression of the heterozygous X-linked condition in female. Much more commonly, the normal allele is inactivated in only some of the cells, and thus the heterozygous female expresses the disorder partially. The proportion of defective red cells in the heterozygous females depends on the random inactivation of one of the X chromosomes. However, the severity of the hemolytic reaction is almost always less in heterozygous women than in homozygous men. Now, example of X-linked recessive disorders. Now, I give a table, so I'll be reading out the disorders system by system. So in the musculoskeletal system, an example is the Duchenne Muscular Dystrophy. In the blood, or in the cardiovascular system, you have Hemophilia A and Hemophilia B. We have chronic granulomatous disease. We have glucose 6-phosphate dehydrogenase deficiency. Now, in the immune system, we have agammaglobulinemia. You have Wiskott acute syndrome. Now, in the metabolic system, you have diabetes insipidus. We have Lesch-Nyhan syndrome. Then in the nervous system, we have Fragile X syndrome. There are only a few X-linked dominant conditions. They are caused by dominant disease associated with alleles on the X chromosomes. These disorders are transmitted by an affected heterozygous female to half her sons and half her daughters, and by an affected male parent to all his daughters but none of his sons, if the female parent is unaffected. Vitamin D resistant rickets is an example of this type of inheritance. Now, we'll talk about mitochondrial inheritance. Mitochondrial inheritance is mediated by cytoplasmic genes, cytoplasmic mitochondrial genes, which are inherited exclusively by maternal transmission. So, mitochondrial inheritance is inherited exclusively by the maternal transmission. So, multifactorial inheritance. This then implies the interaction of two or more genes of small effects with environmental factors. Multifactorial inheritance implies the interaction of two or more genes of small effects with environmental factors. Congenital anomalies with multifactorial bases arise as a result of inheritance of multiple genetic polymorphism that confer a susceptibility phenotype. The interaction of this underlying phenotype with the environment is then required, therefore, the disorder becomes manifest. The interaction of this underlying phenotype with the environment is then required before the disorder becomes manifest. So, this genetic disorder doesn't manifest if an environmental factor is not involved. For example of this is a shallow acetabular socket and the laxity of the supporting ligaments are believed to be genetically determined, whereas frank breech position in utero with hips flexed and knees extended is a key environmental factor for the development of a shallow acetabular socket and laxity. Monozygotic concordance rate for some common congenital anomalies like cleft lip or cleft palate is only in the range of 25% to 50%. A good example is marked reduction in the incidence of neural tooth defects by periconceptional intake of folic acid in diet. Thank you for listening. If you find this very educative, please kindly like, share, and subscribe.